Good. Okay. Well, I think we will get started. I think we have a quorum. So, well, first I'd like to welcome everybody to this Donders lecture, and I'm delighted and honoured to introduce our Donders lecturer for today, Professor Anne Churchland, who joins us very early this morning, for her at least, from Los Angeles. Anne has actually had quite a bi-coastal life in the US, crisscrossing the country several times. She got her bachelor's degree from Wellesley College in Massachusetts, and then decamped to San Francisco, where she received her PhD at UCSF in 2003. She followed this with a postdoc in Mike Shadlin's lab at the University of Washington, where she studied the neural mechanisms of decision-making. She then moved back east to Cold Spring Harbor Laboratories in New York as principal investigator in 2010. And here her lab developed multisensory paradigms to investigate neural circuitry supporting perceptual decision-making in both rodents and humans. And in 2019, she was named chair of Cold Spring Harbor Neuroscience. But despite this rather impressive honor, in 2020, she returned to California and is currently professor of neurobiology at the Geffen School of Medicine at UCLA. I don't want to preempt her talk, of course, but her research has been very influential in the areas of multisensory integration and perceptual decision-making. A particular feature of her work has been in developing advances in behavioral techniques to assess these multisensory and decision-making abilities in rodents, techniques which had previously only really been available in primates. So I'm sure we'll hear a lot more about that this afternoon. In addition to her direct research accomplishments, Anne is also a founding member of the International Brain Laboratory, which is a collaborative research group aiming to develop the first global model of decision-making in mice. And she's also an advisor to the Allen Institute for Brain Science. Interestingly, she's also the founder of Anne's List, which is annslist.net, which is a website promoting equality and gender representation in scientific meetings, and is a really useful resource for anyone organizing a meeting or a colloquium series or anything like that. It's I think, a very nice endeavor. Now, while we're sad, of course, that it wasn't possible for her to cross the country and the ocean one more time to be here in person, and we are delighted to hear from Anne today, at least virtually. And of course, we hope we get the chance to visit or to welcome you here to Nijmegen at some point in the future. But for now, she's somewhat heroically been willing to arise and give this Donders lecture at 7 a.m. California time. I can't even begin to imagine being coherent at 7 a.m., never mind giving a lecture. But for this, we're extremely grateful. So just before we start, some logistics. If everyone, of course, can mute themselves, everyone except Anne, can mute themselves for the duration of the talk, that'll help. And if it's possible, please turn on your cameras so Anne has some little smattering of Nijmegen to look at as she gives her lectures. We'll have some time for questions at the end, so please save up your questions until then, and I'll moderate the question session. Okay, so without further ado, please join me in welcoming Anne, and we very much look forward to your talk.
Okay, wonderful. Thank you so much for that introduction, and thank you very much for having me here today. I also wish very much that I could be there in person so that we could have relaxed chats over coffees and lunches and so on, but I'm still nonetheless really pleased that we were able to get together in this virtual format, and especially because it means people can really come from all over and be joining the seminar despite many different geographical locations and life circumstances. So thank you all very much.
We can go down to the front desk.
We have a rogue microphone on. Before I start naming and shaming people, please turn off your microphones. Luke Miller. Oh.
Okay, all right, no worries. So the starting point for the I'm going to tell you about today is a paper that we published a few years ago now about the impact of movements on neural activity. And some of you may know about this work. It's published and may have even worked closely with the video and neural data that we collected and also shared as part of this effort. So I'll start off the talk by briefly reviewing kind of what we did there, and then tell you about the very new direction that we've taken to build off those original findings. And as you'll see, this new direction was sparked by collaborations with molecular and developmental biologist colleagues, and a large body of work in those fields that we were starting might be relevant to us as systems neuroscientists hoping to understand decision circuits. So this work was led by Simon Musil when he used to be a postdoc in my lab, and Matt Kaufman. Both of them now have their own labs, Simon at ULIC and Matt at the University of Chicago. So when we started thinking about this work, we wanted to think about movements, not only as we had done in the past, but also more broadly. And so we reasoned that although we and others in the field had focused extensively on what you might call task-aligned instructed movements, that we hadn't really considered other sorts of movements. So task-aligned instructed movements are ones that we taught the animal to make, say, for instance, making a licking movement at a stereotype moment during each of the number of trials. But of course, there might be uninstructed movements. So perhaps when a stimulus is flickering on and off, the animal moves its hind paw. We never taught the animal to do that, but maybe it does it anyway. And these uninstructed movements might have a task-aligned component in the sense that they occur at a stereotype moment relative to task events, or also might be task independent in the sense that they occur idiosyncratically at different moments in the trial. And of course, instructive movements might have a task independent component as well, because of any of you who have done animal work or even human work, even when you tell subjects to make a movement at a particular time, there's always a certain amount of variability in when that movement actually occurs. So why were we thinking about movements in this way? Well, our big question was this: do brains prioritize cognition and instructed movements, or do other signals dominate? And so to do this, we used an auditory and visual spatial localization task, where it's the job of the animal to detect an auditory or visual stimulus that's presented on one side and to make a licking movement to the corresponding side. And I'll show you a video of that behavior in a couple of minutes. I should say that that builds off a growing body of work that's come out of my lab over the years, where we've tried to borrow tasks from the human perceptual literature and bring them to rodents so that we can study computations that we think are perhaps conserved across very distinct species. One of the most recent ones is listed at the bottom. It's just come out a few months ago in eLife about lapses, when we make decisions and have errors even on the very simplest of those decisions. So if you're interested in that, I encourage you to check it out. On this somewhat simpler detection behavior, I'm going to show you a video of what it looks like. So here you're looking at the mouse from the bottom up. So the camera's underneath the mouse, and you can see its whiskers and its little nose at the very top there. And each trial begins with the mouse making contact with two handles that you see at the bottom there. They move in, and when the mouse is ready to initiate trials, he makes contact with one of those. I'll show you what that looks like now. Handles come in, and that electrical contact triggers the trial to start. And now you're looking at the mouse from the opposite point of view, this time from the back. And you're going to see this happens to be a visual trial, although this mouse will be doing both visual and auditory trials. And you're going to see a visual stimulus be presented 2 times in rapid succession on the right. And this is, again, from the back. So that's the mouse's tail that's hanging down at the bottom of the screen. Okay, great. So after that, the mouse has to make a decision. Hopefully it decided that the stimulus was on the right. Hold that decision in mind for a 1-second delay period. And then after that, it's time to make a response. At this moment, the 2 spouts are going to move in so the mouse can make contact with them using a licking movement. And then the spout that the mouse doesn't choose will move out of the way just to prevent the mouse from changing its mind. Okay, so as you can tell from these slides, we collected a lot of video during this behavior. And we did this for a variety of reasons. We had previously looked at video in a rather informal way, but we realized, and this was really what Simon took the lead on, that having collected all this video data, it put us in a good position to track a number of movements about the animal. For example, we could track the pupil diameter, which fluctuates gradually on a number of timescales, including over the course of the trial. And we can estimate that over time. And we can also track other movements such as the whisking that the mouse makes. And this turned out to be rather important later on, so keep it in mind. Excuse me. So remember that our goal here was to determine whether cognitive or movement signals dominate neural activity. So armed with this new task in hand, we set about trying to figure out, well, where would the activity in the brain possibly answer this question? And we decided we wanted to take a rather broad view. Instead of focusing on an area for which we had a specific hypothesis, we said, let's look across the whole dorsal cortex, because who really knows? There could be any of a number of areas that are relevant to understanding this question. So in order to do this, we built a custom wide field macroscope. It looks a little bit like this. So there are a camera at the top to capture activity that's coming from the brain. So we measure activity from GCAMP, which is a genetically encoded calcium indicator. And for any of you who are not familiar with that, all it means is that when the neuron fires, the neuron turns green, and that emits photons that we can catch with this camera that you see at the top. It's of course very complicated to go from calcium activity to spikes because those two are related in a rather complicated way. And that's something that we think about and take into account for all of our analyses. For any aficionados in the audience, I'll point out that we illuminate at 2 different wavelengths, 473 nanometers, that's to activate the GCAMP, and then also at a shorter wavelength, we illuminate with violet light, and that's to do a hemodynamic correction so that we can focus on the GCAMP signals that are separate from the slower hemodynamic responses. This is kind of a cool technique in that it allows us to see the whole dorsal cortex at the same time, which gives a really, really broad view on decision-making activity. It's also pretty cheap because you can just use LEDs instead of lasers. So if anyone is interested in building such a setup, it's kind of an easy and lower cost alternative to 2-photon imaging. And we actually wrote a protocol about this approach that I've listed below. So I'm going to show you now just what the data looked like as it was coming in. So you can sit there in your seat and kind of see, as we did, the video and the neural data together and kind of take our point of view. And I'll tell you a little bit what jumps out at us about this data. So these are all going to be simultaneous in time, although they're slowed down quite a bit because the mouse moves quickly and the brain reacts quickly. And I'll show you a number of example trials on each of which the mouse makes a decision about a sensory stimulus. So as we were looking at this data, there are really 2 things that jumped out at us. And the first thing that jumped out at us is that the cortical activity is really quite widespread. So it looked like neural structures all over the dorsal cortex were recruited even during these rather simple decisions. So that was a surprise to us, surprise number 1. The second surprising thing is that the activity was quite different trial to trial. So the pattern of activity that we saw across the brain, like here on trial 4, is unique. It's different from trial 3 and trial 2 and trial 1. And to me, that was surprising because of course the animal is doing more or less the same thing on every trial. Naturally, the stimulus is presented on a different side, but there aren't massive differences in what's going on in each trial here. So this was a bit puzzling. So in order to make sense of this single trial activity, we first were a bit skeptical of wide field signals and said, okay, let's just go back to our old friend mean activity here and just make sure that we're getting the right signals in the right place at the right time. So this shows you a really grand average of trial activity for all visual trials at different moments in the trial. And you'll notice that when the stimulus comes on, the back of the brain, that's primary visual cortex, is bright yellow. That makes sense. Visual stimulus is on, V1 lights up. For any aficionados, we pooled left and right trials here, which is why the activity is present on both sides. So this was to a first pass reassuring. And then we said, well, okay, actually, fortunately, the field knows a lot about V1. Let's really focus there and see once again that we're getting reasonable signals, really to kind of put the heels of this technique to the fire and make sure it's really making sense so that we can then go back to these questions about variability and widespread activity that I mentioned a moment ago. So the first thing we said is, okay, well, one thing we know about V1 is that you're going to have a bigger response to lights than to sound. So that's pretty straightforward. So let's look at the average neural activity. That's the delta F over F on the vertical axis as a function of time on the horizontal axis. And we'll look at that for both trials with lights and trials with sounds. So there are 2 traces here, but they're overlapping because before the stimulus comes on, it doesn't matter whether there are lights or sounds, they're always the same. But once the stimulus comes on, of course, you see a much larger response in V1 for lights than for sounds. And it has really exactly the temporal dynamics that you would expect based on the well-studied properties of area V1. We did the same comparison of V1 activity for stimuli on the left versus the right, and again found, as expected, that we had a much larger response in the left V1 when the lights were on the right. And that's, of course, because V1 cells are primarily driven by stimuli in the contralateral hemifield. So taken together, these analyses of the mean, along with the number of other things we did, retinotopic mapping and so on, really reassured us that overall we were measuring meaningful signals with wide field imaging that matched well-established properties of a number of areas in the brain. So then we said, great, things seem to be working. Now let's go back to this question we had about the variability in trial activity and how widespread it is. And our goal at the time was to take for any pixel in the brain, or really sort of a reduced dimensionality version of that, we said we want to be able to predict the activity on each trial. So let's say these are the fluctuations on trial 1 and trial 2 and so on. We want to build a model so that we can train it up on a bunch of data and then test it on held out data and predict these single trial fluctuations. So I have to be honest, when the postdocs, Simon and Matt in the lab told me this is what they wanted to do, I was like, folks, come on, you're crazy. This is not a good idea. Cortical activity is famously variable trial to trial. There's no way you're going to be capture all these fluctuations. And they said, well, you know, that may be true, but let's remember something here. We have a lot of good regressors, that is behavioral variables at our disposal that might potentially account for these fluctuations. And they said, well, here's a couple of examples of them. We have the stimulus that occurs at different times on every trial by experimental design. And of course, things like the handle grab and the licks, they occur at different moments because they're under the control of the animal, who naturally does a different thing every trial. So those are potential behavioral variables that might explain these fluctuations, fine. And then we also had this video data that I mentioned a moment ago. And so we wondered whether there might be features of the video that might also explain these fluctuations. We did a bunch of things with video. I'll tell you more about that as time goes by. But the very first pass thing we did was just to switch the video down to a smaller number of dimensions. You can see the top 3 PCs of activity here for 3 example trials. They sort of look like nothing because they're in an arbitrary coordinate system. And we put all of these variables together in a big design matrix. And then we trained up a model with a ridge regression penalty to learn the weights that would match these regressors to the neural activity. And then, of course, we can test this model on held out trials. In the discussion, if folks are interested, we can talk about the rather different variables that are included in this model. These ones are event-based, things like when the stimulus comes on, whereas these ones vary more continuously in an analog fashion. And we thought deeply about the impact of including different variable types in the model. So if you're interested in that, ask me in the discussion. But the upshot of it is we trained up the model and tested it on held out trials. So now if you look to the right where the trial activity is, I'm going to show you the predictions of the model on trials that we never fit the model to. You'll see that now in red. So you can see that we were able to capture these fluctuations, to predict these fluctuations at the single trial level actually pretty well. This to me was really surprising, and I certainly wouldn't expect this audience to be convinced by 3 example trials. So I'm going to show you now how well we were able to predict activity on cross-validated trials across the brain. So this metric here, CVR squared, that's for cross-validated R squared. So that's just simply the correlation between what we predicted and what actually happened on those cross-validated trials. And we can get a value, of course, for every pixel in the brain. So you can see we were able to capture activity pretty well. We were able to explain about 42% of single trial variability in the fluorescence signal, which was a super surprise to me. I did not think we'd be able to do that well. And this was true at multiple different moments in the stimulus. It wasn't limited to a particular time. So this was great. So this allowed us to say, well, now let's look under the hood, because remember, the real question is, what's driving this neural activity? Is it cognitive variables, movements? What is it? Well, great. Now we have a model that works. Let's look under the hood of the model and see which behavioral variables are really doing the job here. Is it these decision-related variables, or is it something else? So what I'm showing you now here is the amount of variance captured by each of a number of variables. And I put them into groups here. These are what we call task variables, and we can certainly debate these groupings in the discussion if folks have concerns. And so there are things like the onset of the stimulus, whether a reward was given, what happened on the previous trial, the sort of variables that any self-respecting decision scientist would consider. And on the vertical axis, that tells you how much variance was explained by each of these behavioral variables. And, you know, some amount, that's fine. So then we looked at instructed movements, ones that we taught the animal to make, things like licks and handle grabs. And those accounted for as much variance as the greatest task variables. So certainly is a good thing. People have been thinking about instructed movements for a long time. They absolutely matter. So, you know, the good thing that folks have been doing that. Here was the surprise. The surprise was when we looked at the impact of uninstructed movements, things we never told the animal to do and didn't even really know the animal was doing until we looked at the video. These uninstructed movements accounted for more variance than any of the other stuff that we thought was going to matter more. So these include things like pupil movements, hind paw, nose, the body movements, whisking, the video and video ME. That's just the first derivative of the video. It's the motion energy of the video. So basically stuff in the video that we didn't capture with any of these other movements. So as I said, we were really surprised by this outcome and did a number of controls, including a much more conservative test where we only looked at the unique contribution, the delta R squared. But basically, no matter what we did, and here I've pooled these variables within each of their groups, task, instructed, and uninstructed. No matter what we did, the uninstructed movements always account for far more variance than any of the instructed movements. You might wonder whether this is because the uninstructed movements are sort of supporting the instructed ones, right? Like maybe when the animal does the handle grab, the animal kind of lurches forward. So there's an uninstructed movement from the hind paw, perhaps. If that's the case, you would expect the contribution of uninstructed and instructed movements to peak together, or maybe one a little bit before the other. That's not what happens. The uninstructed movements account for more variance throughout the trial, even before the trial starts. And if anything, their contribution dips a little bit around the time of the instructed movements, because it's at those times when finally some variance is accounted for by something we taught the animal to do. So this plot up here kind of unfolds things in time. So I'm just finally going to unfold things in space and time for you here. That's a movie. And here you can just see the contribution of each of these variable groups at every moment in time. In a moment, the visual stimulus comes on. There it is. Briefly, V1 task variables account for some variance in V1. But after that, it's really all uninstructed movements. So you're likely worried about a number of things at this point, and you really should be, because wide field imaging, for one thing, pools the activity of many neurons together. So you might wonder whether perhaps this is just a weird artifact of wide field imaging, or maybe it's the fact that it's a bulk cell measure and not a single cell measure, and there's some strange averaging going on. We were worried about the same thing. So for that reason, we went to a single cell approach. We looked at both 2-photon and also some electrophysiological data using neuropixels that I don't have time to tell you about today. But the short part of the story here is that we replicated these results using single cell approaches like 2-photon imaging. We recorded about 12,000 neurons in 5 different cortical areas: anterolateral motor cortex, medial motor cortex, primary somatosensory cortex, retrosplenial cortex, and primary visual cortex. And I'm just going to summarize all of the 2-photon data here with this plot. So this is a little bit thorny. So if you want to take a 1-minute break for a moment, you can. But if you're kind of psyched about movements and task variables, stay with me, because I think this is a neat visualization. So on the vertical axis, that's this CDR squared quantity, how much unique variance is explained. And the neuron number is on the horizontal axis. And the neurons are ordered here by how much of the total variance was accounted for by the full model. So the neuron at the far left is the one that was like, we knew exactly what the neuron did. We could account for almost all the variance. And the one at the right, we couldn't account for much at all. Okay, great. So that's why it goes in this beautiful kind of gray, the beautiful like smooth pattern. Okay, then for each neuron, we broke up how much of that variance was explainable by uninstructed movements, task, and instructed. So there's basically a few things to notice here. Excuse me. So the first thing is that movements almost always dominate, right? For almost every neuron, the black lines are at the top and the green and blue are below. You sort of knew that already from the other plot, the other analyses, but this replicates it at the level of single neuron data, which is really, really important. The other thing that's really interesting to me is that you can see that neurons are very idiosyncratic, right? So it isn't the case that in every neuron you measure, it's 80% uninstructed, 10% task and 10% instructed. They vary a lot. We found a few neurons that were true task neurons. They didn't care what the animal was doing. They only cared about the decision variables. Those neurons were rare, but they did exist. Other neurons were almost entirely dominated by movements, and a few were a more even mix of the three. So I guess to me, this is interesting because I used to kind of go into the cortex and see a particular pattern of activity, maybe one that resembled my favorite neural computation, and think, oh, great, this is evidence that, brain is doing computation X. The way I think about it now is quite different, that I go in and measure a neuron and I think, okay, this neuron surely has mixed selectivity for a number of different variables. I need to really do the analysis to pull these apart and to see which aspects of that neural response, that PSTH, if you will, are truly driven by the putative computation and which ones are driven by other features. So for me, this was a real game changer in terms of how I look at neural activity. And either way, it certainly demonstrates that at the single neuron level, movements dominate neural activity, even in trained experts. Okay, so I think looking back at that approach, there was a lot that was novel about it. Certainly in our consideration of multiple, quite diverse types of movements, and also in our somewhat hypothesis-free method of thinking about it. Remember, we sort of took all that raw video data and just kind of threw it in the model and said, okay, model, have at it. And this allowed us to realize something about movements that wasn't a particular hypothesis of ours, and that in fact was rather to us was really quite surprising. But in another sense, I think that our approach was kind of a variant of a classic approach in which we measured neural activity without very much knowledge of the cell types that we were recording. Now, that's not completely true, right? So you remember from earlier in the talk, we used what's called EMX-Cree mice. That means that we use a promoter called EMX that labels only excitatory neurons. So we were focusing on excitatory neurons and not inhibitory neurons. And we paid a little bit of attention to layer in some of the analyses as well. But overall, we kind of considered all of the excitatory neurons together as one big kind of almost monolithic group. So looking back on this, it's interesting because at the same time that we and many others were using this kind of approach of pooling all excitatory neurons together, there's really a revolution happening in systems neuroscience. And that's a revolution in terms of understanding and cataloging the diverse cell types, especially excitatory cell types in the brain. So this is priority number one of the US Brain Initiative, is discovering diversity, which aims to identify and provide experimental access to the different brain cell types to determine their role in health and disease. Anyone who was reading Nature last week, this was kind of the big reveal moment for the Brain Initiative Cell Census Network. They had 17 papers in Nature last week because this has really been one of the big successes of the US Brain Initiative. There have been many, many papers about this topic. I'm thinking of different ways to define cell type, either by developmental lineage, location in the cortex, or sometimes both. And interestingly, this cell type diversity among systems neuroscientists is widely recognized for inhibitory neurons, right? So most people know about VIP neurons, PV, somatostatin, and so on. And there have been a lot of papers that are explaining and highlighting the really distinct roles of these different types of inhibitory neurons. We now know that it's not at all right to think of inhibitory neurons as one single monolithic group. These different types do really quite different things. But that appreciation of diversity is only just beginning to extend to excitatory pyramidal neurons, even though pyramidal neurons, if anything, have perhaps more diversity than inhibitory neurons. This diversity, I think, has really been quite overlooked. And so the starting point for the work I'm going to tell you about now, sorry, this is cut off, is that we have on the one hand, extensive knowledge of the diversity among pyramidal neurons, thanks to work from the Brain Initiative and many other groups as well. On the other hand, we have really insufficient consideration of diversity in the experiments that link brain and behavior. And so in thinking about this, people in my lab and others at Cold Spring Harbor, we started to realize that there was at least a gap, maybe really kind of more of a chasm between what had been discovered in terms of cell type diversity and how we were doing our experiments as systems neuroscientists. Now, I don't want to overstate this too much. There are some papers that look at like a single area, maybe with 2-photon imaging, occasionally with electrophysiology, and say, what do the different pyramidal neurons do within that single area? Super important work. But what we realized is because of our expertise in wide field imaging, we were in a position not just to look within a single area, but to look really broadly and to see whether the putative differences among pyramidal neurons that had been identified in a single area, to see whether those generalize across the entire dorsal cortex. And wide field imaging is really the perfect technique to get a big cortex-wide view of what's happening. And so to do this, we reasoned that we might take advantage of developmentally defined cell types and get this broad view of how they contribute to decision-making. Okay, so the big question here is, do specific cell types, in this case, pyramidal neuron types, make distinct contributions to decision-making circuits? So I'm going to focus on a couple of different pyramidal cell types here. So I'm going to tell you what they are, and then I'll use the same pipelines as I did before to probe their specific contribution to decision-making and see if they all do the same thing or whether they do different things. So the first pyramidal neuron type are called FESF2. I'm going to call these PT for pyramidal tract neurons. These are defined by FESF2, which is a transcription factor. It's kind of a developmental regulator in regulator during development, really key for patterning the developing brain. It's expressed throughout the cortex in quite a uniform way. So those white, whenever you see a cell being white, like those ones in deeper layer 5B in the cortex, that means it's a FESF positive neuron. You can see that they're everywhere from front to back, and they're always in layer 5B. They project to a bunch of different targets, but primarily subcortical, to the superior colliculus, to the pons, to the cortical spinal tract, and so on. Okay, the second class I hope you'll keep in mind are plexin D1. That's another developmentally defined cell type. I'm going to refer to these as IT for intrathelencephalic neurons. These neurons also play a role in development a little bit later than FESF neurons. They're located a little bit more superficially in layer 5A, and also some of them even quite superficially in layer 2-3. You can see again, those white dots are the cells. I'm not sure my movie's going to play here. There it is. And they're expressed pretty uniformly from the front to the back of the cortex, although their distribution is slightly less uniform than FESF within a given section. Well, where do they project? These ones are a little more complicated, and this turned out to be a bugaboo for us, so keep it in mind. They actually project to two different places. The IT neurons have a big cortical projection, so they project to cortex on the other side. And then they also have a big projection that goes corticostriatally. So this to me is kind of neat because this is a cell type that's defined by what happened, long ago when the mouse was like a little baby mouse, but they still exist in the adult animal, but they have two different projection targets. So we kind of pondered this and said, well, how should we think about this? On the one hand, this is one cell type if you look through the window of development, but if you look through the window of laminar position, well, maybe they're two different cell types. So we grappled with that, and I'll return to that issue later in the talk. We came up with a way to kind of tease apart those contributions, but this kind of highlights that as with many aspects of the field, and I think cell types in general, it's often hard to know what constitutes a cell type and which cells to lump together and which to pull apart. Okay, so to summarize what I've just told you, I've described two classes of neurons, PT, which mostly go subcorticofugally, they go down to the spinal cord, colliculus, and so on, and IT, which project to the striatum in the cortex. So fortunately, as a comparison point, we also have the same neuron, the same type of neuron labeling method that I told you about before, EMX, and that just means all excitatory neurons are labeled. And so what we decided to do was to take mice where either all the neurons were labeled, the PT neurons were labeled, or the IT neurons were labeled. And we did this in collaboration with our colleague Josh Wong at Cold Spring Harbor, who made the mice. And then taking each of these mice, look at their activity in the wide field to see what PT versus IT neurons do during decision-making, and to compare that to mice in which all excitatory neurons are labeled. So we had two key questions we wanted to ask here. Do PT and IT cells exhibit unique neural dynamics? And do PT and IT cells play distinct functional roles? So I'll start with this first question here. We used a similar task to the one that I told you about previously, except it has an added layer of depth, which is that now it's a discrimination task. So rather than just having, let's say, lights on the left, there are both flickering lights on the left and the right, and it's the job of the animal to go to the side where more clicks or more flashes are present. So this is rather more challenging for the animal in the sense that they have to make a discrimination between two different sensory streams that are coming in. And we also sometimes use the detection task as well. So you can see the psychometric functions, the proportion of right choices as a function of the number of clicks for the discrimination task and also the detection task. And on the left, you can see their performance improving over many training days. So we use the same approach as before, wide field imaging, to measure the activity of these cells, and the same pipelines that I described earlier in the talk to separately look at the contribution of different cell types to things like task variables, movement variables, and so on. Okay, so before measuring neural activity and decision-making, as before, Simon, who's generated retinotopic maps, he was again the leader on this project, along with Richard Sun, who's another former postdoc from my lab. And these retinotopic maps allow us to identify primary visual cortex and usually around 5 or 6 surrounding areas. This is a great thing to be able to do to verify that the system is working generally, but also was very reassuring to us in PT and IT neurons, because of course we worried, well, hang on a sec, like with PT neurons, those cell bodies are deep in the cortex, they're layer 5B, we might not be able to see anything at all. This was really reassuring because it tells us that although we need to think very carefully about comparing activity across cell lines, we can get really good, meaningful signals within each of these different mice. Okay, so having established that we're able to measure neural activity reasonably in all 3 of these mice, now it was time to look at what they did during decision-making. And I'm going to again show you movies similar to what I showed you before, but this time you're going to see decision-making activity through the lens of 3 different cell types. You have all of them together in EMX on the left, just the plexin or IT cells, sorry, I should have relabeled that in the middle, and the PT FESF2 cells on the right. So there's only one thing that I hope you will remember from those videos, which is that they are not the same. So in decision-making studies where we lump all excitatory neurons together, we're mixing together signals that are doing rather different things. And that was clear to us from the moment that we looked at the movies. We could tell immediately that, of course, they weren't the same. But we really needed to dig deeper to explain exactly how they are not the same and to relate those differences to decision-making activity. So that's where the real work began. So the first thing that we did was to say, okay, just forget about these, decision-related task events and all this. Let's just look at the activity really broadly and just kind of see how complex it is. Just look at the overall spatial patterns that are occurring across the brain with each cell type and compare them to each other. So the first thing that we did was to use semi-non-negative matrix factorization, which is just a way of breaking down a matrix into spatial and temporal components, and to see how many dimensions we needed to account for 99% of the variance in each cell type. So what I've shown you on this plot is the number of components needed to explain 99% of the variance for each of the 3 cell types, so measured in each of the mice. So there are 4 mice in each group, or sometimes 5. Each mouse is shown separately, and because there are multiple sessions, there are multiple points for each mouse. What I hope is clear here is that very consistently, we saw that PT cells were much lower dimensional. So we needed many fewer components to explain the majority of the variance for PT cells. So to put that in just in everyday language, it means that the overall spatiotemporal patterns are just kind of simpler. They're sort of less complicated. So we then wanted to see how these spatial components related to each other. So we took all these components and embedded them in this 2-dimensional space using UMAP, and that's what you see here. So each point is one component from one animal on one session. So that's why there are so many of them, because we get quite a lot of components for each session, and we have a lot of sessions and 4 or 5 animals. And what was clear to us looking in this 2-dimensional space is that these points tend to really cluster together. So what that means is that components within a particular cell type like PT, they tend to be rather similar to each other and distinct from components that come from IT or from EMX. So what that means is that the patterns of activity that you see across the brain, they aren't just simpler for PT neurons, for example, they're actually fundamentally different. So each of these cell types is driving different spatiotemporal patterns or spectral temporal patterns across the brain. Now, you might worry about this a little bit because perhaps there are just certain chunks of the brain that are missing, right? Like maybe we didn't get good expression in one part of the brain for one cell type. And so that's why, for instance, PT cells are lower dimensional. Well, to get at that, we used a technique developed by Shreya Saxena when she used to be a postdoc in Liam Paninsky's lab. And she developed a technique called LocaNF, which very similar to SNMF, to semi-negative matrix factorization, except that you seed the algorithm with particular parts of space, in this case with the Allen Institute Common Coordinate Frame. And this allows you to look within particular regions how many components there are. And this is really useful for the question I just mentioned, because if, for example, a particular region is missing, we'll be able to see that with LocaNF. But instead, what we found is that we actually had really similar number of components for each of the cell types in LocaNF, and also that these tended to form cell type specific clusters as well, and that the clusters that were really dominated by a particular cell type, like this one you see here, specific cluster 1, SBC1 and SBC2, tended to be very small, kind of within Allen Institute regions, whereas clusters that tended to have very diverse cell types tended to be larger, kind of like super regions. So this suggested that there might be some structure beyond that we see with the Allen Institute CCF that's driven by cell type specific activity. Okay, so the first answer to the first question, do PT and IT cells exhibit unique neural dynamics? The answer is absolutely yes, they do. Do PT and IT cells play distinct functional roles? So having established that these two different cell types have quite different patterns of activity across the cortex, we wanted to know what they did in relation to decision-making activity. And here we were able to use the pipeline that you're now all pretty familiar with, where we try to explain the single trial variance across the brain. And now we can just extend that to each of these specific cell types. So we repeated the analysis that I told you about earlier in the earlier in the talk, looking at the contribution of movement and task variables. And we found that, as before, movement variables dominate neural activity, just like we've previously seen. That is also true when you just look at IT neurons, and also true when you just look at PT neurons. Again, movements dominate neural activity, no matter what cell type you look at. Okay, so then when we looked at the spatial profile to account for variance, and now you're seeing this delta R squared, this more conservative measure of accounting for neural activity. You can see this now separately for movement and task variables. We found that although it's true that the cells have in common that movements always dominate, that there were big differences in the extent to which task variables explained activity in different areas. So just to give a couple of examples here. So the choice variable tended to explain activity well for PT neurons, but not nearly so much for IT neurons, whereas the previous success didn't account for much variance at all of PT neurons, but a lot more for IT neurons. And similarly with the stimulus-driven response. So this kind of hinted to us that even though these cells had something in common in terms of movements dominating neural activity, there also might be some really interesting differences among the neurons as well. So to understand better what those differences are, we looked at the temporal event kernels that are learned by the model. So remember, the job of the model is to fit a kernel that's varying over time to explain the variance. And we can look at those kernels and see, first of all, how big they are for different in different areas, and then also look at their time course. So this is now the stimulus kernel. So this is what's learned by the model to explain the stimulus-driven activity, separate from every other variable, movements and so on. So this is a very pure stimulus representation. And this was kind of cool. So this is quite similar across all three cell types. So for all excitatory, IT, and PT, we see after the stimulus comes on, there's a fairly stereotyped a bump of activity that then kind of often peters out, sometimes lingers around a bit longer. That was true in a number of different cortical areas and was really common across the three different cell types. For me, the coolest part about this was looking at the PT neurons, those are over on the right, and noticing that around the parietal cortex, that they had this really strong stimulus-driven, or at least activity around the time of the stimulus. And the reason I thought this was intriguing and kind of surprising was that PT neurons, they're projecting down to the corticospinal tract, the colliculus. They're sort of thought of like the movement neurons, like they're just there to make you move. And that's what really previous work that's focused on PT neurons has mostly uncovered. So this to me was kind of different because this is in parietal cortex, which is not, I mean, there's movement signals there, but it's not the motor cortex. And we're seeing this activity right around the time of the stimulus, not really a time when there's a lot of movement planning in terms of executing the choice. So this was kind of a first hint that these PT neurons were doing something that was rather more complex than what we might have thought originally. And here's just to zoom in on that area. You can see with the PT neurons, there's this really nice hot spot that's kind of a little bit medial to area AM. We also looked at the ability of the temporal event kernels for choice, right? So this is from the encoding model, the choice signal, and again, found some interesting temporal dynamics, but we took these with kind of a grain of salt because the choice activity is really, really, really small, right? Because most of the activity is driven by movements in the encoding model. The choice component is very small. So though we saw some interesting activity in secondary motor cortex, we decided to switch gears and to train up a decoding model to instead start with the neural activity and try to predict what the animal did. So kind of the opposite of an encoding model. And we found that we were able to decode the animal's choice quite well with all 3 cell types growing over the course of the trial. And what was interesting is when we then went to look at the temporal event kernels, this time for the decoding model, to focus on the frontal cortex and to compare them for EMX, IT, and PT, we saw something kind of interesting, which is that the EMX and PT, those values grew over time, indicating a greater ability of ours to decode a choice as time goes by, a greater importance of those areas to contributing to the choice decode. But what was kind of surprising is this declining role of IT neurons. It's almost as if we needed them less and less, and sometimes they even went negative as time went by in the trial. And that was really quite different from the PT neurons, where their contribution in the frontal cortex really grew as a function of time. Okay, so just to tell you what we found so far, that PT and IT cells drive unique neural dynamics, that PT neurons are active during stimulus presentation, and that PT neurons and IT neurons exhibit different choice-driven activity in M2. This is what I just showed you a moment ago. But this is kind of highlights this problem that I mentioned a few slides ago, which is that you see a decrease like this, and you wonder, is the declining role of choice in IT neurons, or their declining contribution to the choice decode, is it driven by these superficial cortical neurons or the deeper corticostriatal neurons? We can't tell that because with the IT neurons that we use that are developmentally defined, the cells project to both places. So we came up with a new strategy to target the corticostriatal neurons specifically using an anatomical strategy where we expressed CRE all across the cortex, flex GCamp all across the cortex. Sorry, I just said the opposite of what I meant to, and then deliver the CRE to those neurons by making an injection in the striatum with a retrograde transporter that will deliver CRE to the cortex and unleash the GCamp only in those neurons that are projecting to the cortex. Here's an example of what that looks like. You can see we get really nice expression only in layer 5A, not in layer 2-3, labeling only those neurons that are projecting to the striatum. Repeated the pipeline I've been telling you about, verified that we saw the same stimulus activity, and then we're able to make the comparison that I told you about before, looking at this declining contribution to the choice decode, and found that these corticostriatal mice, just like the IT neurons, had a declining contribution to choice. In fact, it was even like that decline was even a little bit steeper. So this verified that there's a really interesting difference between PT and IT neurons around the time of the choice. And also, as I mentioned previously, that there's an interesting role for PT neurons in the parietal cortex. But we really wanted to know about the causal role of these signals, right? These models are a little bit separate from real life. We're encoding things and pulling out the temporal event kernels. We really wanted to go in there and disrupt the activity in a cell type specific way and look at the impact on decisions. And this will be the last thing that I'll tell you about today. So for the IT and PT neurons, the developmentally defined, we injected Excitatory, which is an inhibitory opsin that's activated by blue light. We had to do things slightly differently for the corticostriatal mice so that we could express the Excitatory and the corticostriatal neurons specifically, and then verified that in the EMX mice, that there is causal relevance of 2 candidate areas, a parietal cortex and frontal cortex, and then V1 as a control. So we first verified that these areas that had popped up as interesting in our encoding and decoding models, that they had a causal role with EMX mice, so with all excitatory neurons. We did. That value on the vertical axis is performance. It means that it shows you how much it's impacted by shining the blue light throughout the trial. We see no impact for no light, no impact for V1 suppression, but a big impact for frontal and parietal suppression. So that validates that these frontal and parietal areas are relevant for decision-making generally, allowing us to go to a more granular approach where we suppress at different moments in the trial for each of the different cell types in the 2 areas. So this is kind of cool because we're doing optogenetics, but in this cell type specific way. And I want to highlight really one main thing that I think dovetails really nicely with an earlier finding that I described to you, which is that we see an impairment, that's the quantity on the vertical axis now, when we suppress PT neurons in the parietal cortex during decision formation. So we used to think of these cells like movement cells, PT cells, they're moving the body around. But here we're finding a more complex contribution of these cells because they have a big causal role, even bigger than IT neurons, in the parietal cortex long before the movement's made. And in fact, their role decreases as you get closer to the time of the movement. So this really fits well with our previous observation from the encoding model that PT neurons have a nice and surprising stimulus-driven response and suggests that they might play a role in decision formation that was unknown previously. So PT neurons are not only active, they are active and causally relevant during decision formation. And then finally, by stimulating in the frontal cortex, we demonstrated a large role for PT neurons there as well. And this really kind of replicates previous observations that PT neurons have a large causal role in M2 near the time that the animal reports its choice. So by doing this cell type specific optogenetics, we were able to go beyond the encoding and decoding models and demonstrate a causal role for all of these cell types at specific moments in the trial and in specific places in the brain. Okay, so I've told you so far that PT and IT cells drive unique neural dynamics at both a cortex-wide and a within area scale. That cortical inactivity and optogenetics reveal distinct functional roles. Surprisingly, parietal PT neurons had a strong causal role during decision formation. That was really surprising to me. And finally, that in frontal cortex, all PUINs were required for accurate choices, but showed distinct choice tuning. And this is this interesting declining role of choice in the IT neurons. So the overall take home is that rich cell type specific cortical dynamics shape perceptual decisions. So I'll stop there. I want to thank the people that did this work. The second part of the talk was led, well, both parts of the talk were led by Simon Usall. The first part was co-led by Matt Kaufman, and the second part co-led by Richard Sun. We're also really grateful to the people that have supported our research and made it possible to do these experiments. And thank you all very much for being here today.